Good Hassan, please begin.
Hello everyone, my name is Buddhabhushan. I am third year PhD student at ISOPOPAL in the Department of Biological Sciences. Today I will be presenting the study which is titled with concurrent loss of selected genes in W93 and CFAP46 in pathological response, which we published last year in RSOP. So why we focus on these genes, W93 and CFAP46? First, I will start with this, why we studied these genes. So as we know that host has defense mechanism, that is mucociliary clearance of. So this mucociliary clearance is important to clear the inval pathogens such as viruses, bacteria, and fungi, and even environmental pollutants. So to clear that pathogenic infection, so mucociliary clearance has two main components. First is motile cilia, which move back and forth and clear these pathogens. And other thing is mucus that is secreted by globular cells and other cell types. So that together both helps to clear these pathogens. So as host has this defense mechanism, and opposite, there is continuous arm race going between these viruses. So virus also target this mucociliary clearance. So here you will see that the most of the respiratory viruses are targeting this mucociliary clearance, and they decrease the uh, velocity of this ciliary beating. So here you will see that except some few viruses, almost all the respiratory virus targeting this mucociliary clearance. Moving further, where that genes that we are studying that resides. So this mucociliary clearance of that beating of cilia is mainly achieved via the central apparatus that helps in the beating of cilia. So it is made up two asymmetric pairs of microtubules, that is C1 and C2. So again, this each microtubule has different kind of projections, that is C1A, C1E, C1C, C1D, C1F. So the study of interest of our genes are residing this C1D. So here they are like this. FAP46 is another name for this CFAP46, and this FAP297 is another name for WD93. So this C1D has important role in the speed of that cilia beating, and it affects the waveform in the CRNATs. It also implicated in the primary cell dyskinesia. So, and apart from these two genes, there is another genes that FAP221, FAP454, and CAM1. These are also part of the C1D complex. And the major role of this C1D complex is they potentially reinforce that elasticity and stability of that spin. Moving further, why we look specifically at this W93. So here you will see that upon the influenza infection, this W93 gene is there, so whose expression get modulated. Upon the high pathogenic infection, it get downregulated, and upon low pathogenic infection, it get upregulated, which was found by Kumar and Loeb. So this is brief about methods. What we did, we studied more than 500 vertebrate species spanning from basal to higher vertebrate, spanning the different orders and classes. So what we did, we checked for the presence absence of these two genes in the vertebrate species. We generated multiple sequence alignment using their one-to-one ortholog sequence, and with the help of phylogenetic tree, we did the molecular analysis and selection pressure on that each branch. We also look for the translational titers, whether these genes are expressed in that species or not, using the publicly available RNA datasets. So let's jump into results. So this is the phylogenetic tree, species tree. So this represents 23 key representative species who has the good quality of genome assemblies. So here you will see that W93, and this is Centenny for W93. So here you will see that most of the species of this right flank of the left flank has the PX11A, PLIN1, and KIF7, whereas on the right flank, the MSP1, MSP12, and ANP genes are there. And interesting to note that is in the sauropsis, right flank Centenny got changed. There are different set of genes which are present on the right flank. One more thing to note here is, here you will see that Gallus gallus, which is chicken, shown in the dotted phylogenetic branch. So here you will see that this gene is missing. So here, this missing gene could be possibility that it could be assembly artifact. Maybe genome assembly might not be correct at that locus, and hence the annotation could be missing. So to rule out that possibility, what we use long read data sets from PacBio as well, and which is shown in here with the orange, and nanoproteins, which are more than 10 kb. And we look for whether they are spanning from flanking genes, that is KI1 and PX11. As I mentioned, these are sentinel genes of this W93. So they were continuously spanning from right flank to left. So it ensures that there is no assembly artifact. Then moving further, this gene loss can be compensated by the other protein which has shared domain with this. So there was another protein NDFSO4 which shares this domain with this W93. But based on the clans analysis path we found, they have high sequence divergence. So this again rule out the possibility that this gene get compensated by other gene. So doing the study, comparative genomic study for gene presence subsets of this W93. So what we found that this species has acquired in-frame stop codons in the first exon of this gene, and also there are in-frame stop codon in the 14 and 16 exon, and also there are splice site changes which are shown here. So AGG got converted to AA in the 5' side. So there are also, apart from this, there are complete exon loss, which is shown in the red boxes here, and also partial exon losses are shown with the orange. So here you will see this red branches. This whole is represent the galliforms. Apart from these two, peniopelet and electrotomy, there it's intact. So here it lost. Again, apart from this galliforms, the geese are there. So these 4 species of geese are there where this gene got lost. So, and even based on the shared events. So let's say here Thunderbolt, purple Thunderbolt, which represent that exon 4, 11, and 13 and 14 got lost. So based on this, we make the chronology of events, how this exon got lost, and we estimate that the timing of this gene loss in galliform is around 65 to 46 million years ago. Then when this gene got lost, the ideal there should be no expression. So we check publicly available expression datasets at that locus. So here you will see that PX11 and MIN1 are the flanking genes, which are shown here, the red boxes. And this is the W93 here. So here you will see this top order has this intact gene, and green anolis, ostrich, then again zebrafinch. Here you will see the peaks are going up. So it means their expression is there. But if you see, and again mallard, which is close anseriform and close to galliforms, there also expression is there. But if you see Anser signodus and gallus, so there is no expression for this gene. So it supports that there is loss of this gene, this galliforms and geese species. So this is locus specific expression status. Again, you will see that there is clear line which shows there is no expression in the gallus, also in Melagris and Fascinaceus. The bottom panel all are galliforms, and the top panel shows where this gene is expressed. So apart from this galliforms and geese species, we also look at the other birds where this gene got lost. So one is rifleman, where the 5th exon has in-frame stop codon. So here, this is short read variation of it. So here you will see that the GA gene, which got converted to TAG. So it make the in-frame stop codon. And apart from this in-frame stop codon, there are multiple exons got lost in the terminal portion. And so this gene got pseudogenized in the rifleman. Apart from this, rifleman druff also has in-frame stop codon in the 1st exon and 15th exon. So they also acquired the same frame restricting stop codons by using converting CGA to TGA and AG to TAG. So apart from this, the Anna hummingbird and the speckled mouse birds were the species where this gene got lost. So in the Anna hummingbird, there are also 1 frame deletion and 2 base pair insertion in the exons of this W93. And speckled mouse birds also shows the multiple events of frame restricting changes. So which proves that the gene got lost in this species. So moving to next, as I was while introducing, I mentioned that this gene belongs to C1D complex. So this C1D complex has another gene, that is CFAP46. And there was study which said that there is CFAP46 is directly interact with W93. So we also then after W93, we checked what are the other genes, whether they are intact or not. So based on this, it views that it should be lost because CFAP46 directly interact with W93, and then the FAP46, that is CFAP46 got deleted, then it does not assemble the C1D projections. So, and these are the different scenario. which they are proposed. So this is a scenario where when the FAP46 got mutated, there will be no this 221 and 87. This 87 is another name of C1d87, that is WD93. So if that got mutated, then WD93 will be there, not there. So interestingly, what we found is like wherever there is W93 got lost. So these are the species of where W93 got lost. So these are all the galliform species: chicken, turkey, cotton japonica. The third panel shows all the geese species where this gene got lost, and the other independent species. So this also shows that here also that gene C546 got lost. That is concurrent with the W93. And wherever the W93 was intact, there is no segmental deletion. Here, whenever there is gap, it indicates that there is segmental deletion. And the straight line shows that there is no segmental deletion. And the synteny was conserved at that. So we estimated the size of this deletion based on the mallard. And what we found is that deletion size around 81 kb in the galliforms. And the geese, the deletion size was slightly higher, which are shown here with the blue. It was around 92 kb. So it is huge deletion in the galliform species. And also similar, the deletion was found in the other independent species, but the boundaries are not shared. You will see here the boundaries are shared. Here again, geese also shares the boundary, but in the independent species, there is boundaries not shared among them. So what are the uses of this study? So as we know that there are various unknown viruses, missing viruses, and unknown zoonoses are there. So here if you see in this figure, so these are the reserve species. For example, duck and chicken is prominent example of influenza. So here you will see that duck is carrier or reservoir species for influenza virus, and chicken is most susceptible species of influenza. But there are limitations to study that zoonotic spillover because there are restrictions in viral access to wild animals. There are also difficulty in the biological wet lab tools. But nowadays, more and more genome assemblies are available and computational tools are available. So comparative study can be possible. So here what we see the pattern of gene loss. So mallard, which is resistance. So there is this gene is intact, but where the geese are sporadic mortality and also chickens are also show susceptibility, there the gene got lost. So now the question about what are the other species? Are they susceptible to influenza? And they can be treated in near future. So that need to be studied. So apart from this susceptibility, the study of cryo, they proved that the C1d projections affect the waveform and it implicated primary cell dyskinesia. So what we found that where the gene was intact, so there is transport speed of mucociliary, that pitting is higher compared to turkey where this gene got lost. Again, the wavelength got difference in these two species.
And
So
just to notice that you're at 12 minutes.
Thank you.
Or 2 minutes
This is the conclusion slides. So we established that recurrent and concurrent loss of C546 and W93. So we study approximately 500 species, and it shows widespread conservation apart from this. Birds, there is lack of expression wherever gene got lost. The species which identified that can be used as a natural knockout to study further. These are the future directions. As the previous study shown that it will not form the C1d complex. So cryo study on this gene loss species can be helpful to further strengthen whether there is C1d complex or not. Then there are multiple tools nowadays coming which analyzes the cilia function. So this species identified by our study can be used as a test set to test that tool, whether that's detecting or not. Then also it helps in the zoonotic risk prediction. This is the timeline in the development of this field. So this is the FAP46 mutant, then implication of W93 in influenza. Then this found that C1d complex it is part of C1d complex. And this is our study, which published in the Royal Society of Open Science. So here we establish that this gene are there in this species and it is not there in this species due to segmental deletion and frame disturbance. With this, I would like to acknowledge my PI, Dr. Nagarjun for his constant guidance support, Amia, who was the part of this study, CSR for funding, and my Institute for Infrastructure. And I also would like to acknowledge for this evolution in 2024 and SEC for platform and support. Thank you, everyone.
Thank you. Excellent. So there's no time for questions, so we should move on to the next talk, which is Barbara Mabel. Barbara, you can begin when you're ready.
Okay, thank you. Hello. So the work I'm going to talk about today is very much a team effort. It involves undergraduates, master's students, postdocs, technicians. And what we've been trying to do is develop methods for mixed community analysis. And so what do I mean by that? So mixed community analysis, there's a lot of different ways we're approaching it. So environmental DNA, iDNA for insect DNA. And there's a lot of excitement in the biology of community, obviously, of what we can do with these new sequencing technologies. So we can have environmental samples from various sources. We can do diet analysis using difficult types of substrates like feces, pellets, swabs. We can use blood-feeding organisms like insects, leeches, or vampire bats to sample the vertebrate hosts from their blood meals. And there's even some new studies suggesting you can use what carrion flies carry on their feet for biodiversity surveys in relation to various questions. But there's a lot of challenges with these. So if you're using amplicon-based approaches, primer design is critical, but it's really important that you can test it on a local set of samples to make sure it's going to work. Resolving targets, if you're not using amplicon-based approaches, are challenging. So if you're using deep sequencing approaches, and then the bioinformatics of either approach is critical that you have a reference database that's going to mean that you're going to be able to interpret what you find. So the first example I'm going to talk about is a diet analysis in predatory seabirds. And this is led by an undergraduate, some technicians, and some master's students, but in collaboration with Amandine Gamble. And Amandine is doing a study in the Falkland Islands, where she's doing some disease surveillance of endangered seabirds and marine mammals. And what she asked us to do was whether we could add to her study by analyzing the diet to see who's eating who. And the reason this is important is because it's really complicated. So skuas and caracaras are the main predators, but they have really broad diets. They eat almost anything they come across. And so they could be involved in transmission of disease. The other unknown, though, is the role of rodents. So they also eat rodents, but rodents are also thought to be important for disease transmission. And so in the Falkland Islands, the government has had an eradication program for rodents. But what we don't know is if you take away the rodents, does that mean that these birds are going to eat more seabirds like penguins? And so Amandine wanted us to see if we could test this on her samples. So traditional diet analysis is time consuming because you have to be able to identify the prey items. You can do this from pellets and things, but if you can do it using a DNA barcoding approach, it could be more efficient. And so you do traditional Sanger sequencing and cloning, or you could do short read or long read sequencing to get higher depth. But whatever method you're using, the processes are having the samples, your DNA extraction methods, choosing the primers for amplification, if you're using that kind of method, the type of sequencing, and then the bioinformatics to interpret what you find. And so the importance here, as I said at the start, is the reference sample set is really critical to make sure that you're getting representative amplification of what you think might be in your sample. So the aims of the study that was led by Eve was to optimize a protocol for diet analysis of these birds using a set of positive test pools from local species that were collected on the islands, and then use this to create a comprehensive reference database to ground truth the primers. And then ultimately what we wanted to do was to analyze the diets of these top predators on different islands with and without rodents. And so the first thing we did was the DNA extractions. We had some cloacal swabs from the predators themselves, and then we had some positive control samples from various local species, and we added other things like fish and seals that we had from our local collections. So an important thing to keep in mind here is that we had tissue samples from things that were from Scotland, and we had FTA cards from the Falkland Island species. And I used an amplicon-based approach. We chose a set of primers that had been well tested across a range of vertebrates that were developed by McInnes in 2021. And then we verified them with sequencing. So we found that everything amplified that we put in, we sequenced them, and then we cloned them to see what we were getting out of these mixed samples. But we were particularly interested in seeing whether we could use long read sequencing using MinION, using Oxford nanopore technology because of the potential you could use it as a field tool. So you could take it in the field and maybe diagnose the diets in real time when they're analyzing the samples to collect. So when we looked to do this, we started off with artificial pools. So we created pools from these positive controls. So we started off with 2 sets of samples that were relatively simple, where we had seabirds, penguins, seals, and rodents. And we had, just to see if we could recover things in proportion, we had less seal DNA than we had the others. And then we had some more complicated pools where we had equal amounts of different types of groups. And so when we look at, do the various types of analysis, so this is looking at clones, we only recovered seals and rodents. And the same was true when we used Oxford nanopore sequencing, which should allow more fine-scale resolution. When we did an analysis using the more complicated sets, again, we had this really strong bias towards seals and rodents in the samples. And this was using this original set of primers, and then we used another set that were designed for marine vertebrates. And it was very similar. There was more seabirds and penguins, but overall, we've got some severe amplification biases, especially because we know that the seabirds eat other seabirds. And so it might not be a bad thing if we weren't getting seabirds, if we weren't interested in those, but that's a major part of the diet we were looking for. So when we looked at the real samples, they were dominated by the predator DNA system. So we know that the primers amplify using the predator DNA. And when we cloned them, we only found the predators. And using the standard pipeline, which was an AmpliSeq approach, where we were taking contigs and blasting them, we only found the predators, but we decided to search through the garbage bin. And we took the unmapped reads and did those by hand and assembled them. And we did a bit better. So we could still see mostly predator DNA, but there was more of the prey items that we could see. But when we looked at what they were, remember that these are very low numbers of reads because these are ones that weren't at high enough depth to be assembled into contigs. So we could assemble them to families rather than to species in most cases. But there's a lot of birds in here. And so we know these birds are eating a lot of other birds. So even though we could detect when they're marine mammals and rodents, but if we really wanted to know whether the rodents were making them eat more seabirds, we would have a bias using this kind of approach. So the next example, we've been using tsetse flies to sample mammalian DNA. And the reason tsetse flies are interesting is they're very easy to sample. They're at high numbers. They feed on a very wide range of vertebrates. They're not like mosquitoes. It's both males and females feed, and they feed constantly rather than just before they breed. And so there's a potential you could use them as kind of samplers of vertebrates. So flying syringes, they've been called in other studies. But what we wanted to do that was a bit different was not just to identify what they were eating, but whether we could get whole genome scale data from these mammalian hosts. And this would allow non-invasive sampling of really difficult to sample hosts. So it's not very easy to get close to a buffalo or a hippo. And so if you could use the flies instead, that could be more efficient. Or highly threatened hosts that really want to have a non-invasive method of sampling. And so again, as in the other studies, we wanted to do a test pool first, but in this case, we took DNA that we'd previously done blood meal analysis on, and we'd use cytochrome B analysis, and we resolved the dominant hosts. And these were sampled, we were sure of the dominant host, and we combined them artificially into pools of various types to see if we could recover what the flies had eaten. So some of the pools had only buffalo, some had only elephant, and others were mixed samples. These was all wild samples. These ones in orange include domestic animals and humans. If you've ever encountered a tsetse fly, you don't let it bite you for very long because it's very painful. And so there's a difference in behavior of the host that we were curious about as well. And so when we did shotgun metagenomics using both short read and long read sequencing, what you can see here is that, so here, these were supposed to be elephants, and we recover those quite well. There's a bit of noise in the background, but the buffalo are not recovered well in these samples. And so there's a lot of noise here. And it looks like there's a lot of other things amplifying, but these are also samples where we see variation in the amount of tsetse DNA. And the other thing, warthogs are also overrepresented. So we had an overrepresentation of warthog and elephant DNA, and an underrepresentation of buffalo. And so because there's actually less DNA overall in these pools that are buffalo compared to elephant. And this could be because of the behavior. It could be that the buffalo are swatting the flies away, so they're not taking big blood meals. And so this is another complication with these kind of studies, is there could be difference in degradation that you have to consider, or other reasons why there might be a difference other than just recovery of the DNA. And so what this means, though, is that these shotgun approaches are not going to be useful if you wanted to do an ID or a biodiversity study, because there's too much noise. So we went back to our amplicons to see if we could get higher resolution by using long read sequencing, and also to test whether our primers were really amplifying everything that was in the community. So we had 48 species of mammals that Harriet, my collaborator, had on FTA cards saved since her PhD thesis. And we had sequenced 2 of each species to try just to compare how much amplification we get and whether it's even across hosts. And this time we're using cytochrome B primers that have been developed by Kosher. And when you sequence them for blood meal analysis, so this is typically what it looks like, because if you have more than one, if they feed on multiple hosts, one fly can feed on up to 5 different species in the time you can detect the blood meal. And where they've only fed on 2, you can detect the heterozygous peaks and resolve them. But often people would reject these kind of things, saying they couldn't resolve the blood meal because there's too much background. So when you could clone these to see what was amplifying in these sequences and use MinION, and what we discovered is there's quite a lot of pseudogenes that amplify. And this might not be a problem if there's a database that has them, but not everyone puts their pseudogenes in a database, and is also systematically biased. So all of the canids had pseudogenes, and so we wouldn't have recognized those, and they would have had, because they've got multiple copies, we wouldn't have been able to read this Sanger sequencing results, and so we wouldn't have identified those hosts in the pool.
3 minutes.
Okay, thank you. And there's also variation in amplification, and we found there's mutations in the priming site. So these primers are very general, but they match some species better than others. And we tried redesigning primers, and it didn't make a huge difference, but in a few cases, we got better amplification. So the messages here are, if you do any kind of amplification approach, is going to have biases. In the case of the seabirds, we were using 16S, and so there's length heterogeneity between species. So in this case, the avian amplicons have longer sequences than the mammals, and the mammals were recovered at a lot higher rates than the avian sequences. So it could be that there is more effective amplification in competitive PCRs. But there's also could be related to the quality and quantity of DNA. So in the FTA cards, we had the seals and the seabirds, the seabirds and the penguins were on FTA cards, but the rodents were tissue samples. And so it could be our bias is because of the quality of the DNA, even though we standardize the quantity and quality might be different in the competitive PCR. In the tsetse flies, there could be a difference, as I said, in the behavior. So humans swept them away. So we didn't detect many humans because it could be they didn't feed on them, or, well, because we had them in mixed pools, we didn't recover them. We knew they were there, but the blood meals might be very small. The other thing are these databases and reference sequences. So if you've got pseudogenes or nuclear copies of mitochondrial genes, this could bias your interpretation results. And it's really important that you can make sure that you've got a close match in GenBank. So I'd just like to end by thanking the various people involved. The people over here were involved in the tsetse project. We had some people helping us on the analyses, and these people were involved in the seabird project. So thank you very much.
Excellent. I think we do not have time for questions. So yeah, I should remind people that you can put questions to the speakers in the chat or in the Q&A feature. So our next speaker is Marina. Marina, do you want to share your slides and introduce yourself there? While we wait just a few seconds here.
Barbara, while we wait, there's a question for you in the chat.
And Marina, begin.
Should I? Yes, thank you. So my name is Marina Rafailovic, and I'm a researcher at the Department of Marine Sciences at the University of Gothenburg. And this work is done mainly in collaboration with Alain Lemoine, Rufaria, Chesney Johannesson, and Roger Butler. It is a theoretical study mostly, but just to put it in the context, I wanted to at least show you a visual interpretation of a hybrid zone, what I mean with it. Although for many of you it's probably well known. So on the left you see a picture of a beach, but for a marine snail biologist, it's actually a hybrid zone with marine snails. For example, these Littorina saxatilis, if you go from this along the beach here, you will see a transition from very large individuals to some intermediate ones here in this transition zone or contact zone, and here very small individuals. And this is actually adaptation to the crab predation in this area and actually a strong wave action in this area. So if you plot the phenotype, for example, or if you plot some allele frequencies characteristic to, in this picture, individuals that are small, or we call them wave ecotype, you will actually see these smooth lines that actually we call clines. So the classical hybrid zone theory focuses on the balance between natural selection and migration. And for example, you have an environment which is the geographical area I showed you, for example, but you represent it as a series of small patches. There is some sharp environmental transitions. So for example, on the left, you have selection for small individuals, on the right, selection for large individuals. And as I said, if you measure a phenotype that is under selection, and you plot it as a function of geographical position, actually the mean phenotype, you will see that it has this smooth shape, or clinal shape, with the center at this point of environmental transition. And we also know a very strong theory, or very well-established theory here, is that if we increase or strengthen selection further, but we keep the migration rate the same, we will actually have sharper clines, or actually stronger barrier to gene flow between these extreme types at the ends of the hybrid zone. That's all very well known. What we don't really know what happens, because of course this concerns the expected phenotypes, but what we don't really know is what happens with the distribution of phenotypes or hybrid indices locally, right? So in particular, what happens in the center of this hybrid zone or contact zone, where we see that the average phenotype is really the average between these two extreme types as we expect. So in this classical hybrid zone model, where we have random mating, if we just plot the frequency of phenotypes locally in the center of the zone, we will see that it's a nice unimodal distribution centered around the mean value between these two extreme ecotypes, and these we call actually the true hybrids, right? And this is as opposed to, for example, multimodal hybrid zone multimodal distribution, shown here on the right. You have individuals representing the small individuals on the left, for example, that I call wave ecotype, just to go back to this snail example. On the right, you have a crab ecotype, but nothing in between. So we don't actually have at all true hybrids. So why are we interested in the modality of hybrid zone? Well, for example, it's an indicator of gene flow between these extreme types. It's an indicator of diversity, but it's also theoretically intriguing question. So how do we achieve unimodal as opposed to multimodal distribution locally and vice versa? And as you will see, it's actually quite difficult to escape unimodality. This is not a very new question, of course. It's been around for some time. And for example, this figure is borrowed from Jiggins and Mallet 2000, where they show that there are actually this diversity of modality. For example, this top row is showing unimodal distributions, bottom bimodal distributions, very different species. I will not go into these details, but the modality of hybrid zone can be different with respect to. It can be the same species, but it can just change respectively in dependence of the, for example, sampling location or sampling date. And of course, if I go back to the snails, we also have our own examples. The first species I showed you is this Littorina saxatilis. It's actually, I did not show here, I realize, but it has unimodal local distribution of phenotypes. But the other sister species that actually lives nearby, Littorina fabalis, with some characteristics different, some very similar, it actually has a multimodal distribution, which you actually can see here, but just you have to squeeze a little bit on the side. So the question is really what explains this diversity and how to achieve this in theory. There are, of course, some educated guesses from before that pre-zygotic reproductive barriers are potentially critical to multimodality, for example, sortative mating or habitat choice, or that when we see a multimodality, it's an indicator of a very strong divergent selection. But of course, there is no systematic theoretical studies on this topic still. So what we plan to do here is to actually use an individual-based computer simulation to allow for to turn on and off different potential mechanisms that can contribute, that can change modality of the hybrid zone, and to actually see, okay, so what is realistic, what is trivial, and so on. In particular, we looked at random versus assortative mating. We included or turned off genetic incompatibilities. We looked at different arrangement of adaptive loci, this particularly motivated by the snails examples I gave you, because there we have lots of divergence that is locked in inversions in both species. So we contrast here collinear versus inversion polymorphism, and we also looked at strength of divergence selection. I will not go into details about the model, just it's classical hybrid zone model, but with some other potential mechanisms turned on and off. Just bear in mind that the adaptive trait has a polygenic basis spread across 10 chromosomes. And now I will go straight to the results. So if we look at the collinear arrangement of adaptive loci and we just compare random mating to the assortative mating shown in these two figures here, in all these figures I will actually just plot the distribution of phenotypes in the central part of the hybrid zone, where the effect of multimodality should be actually the strongest, if it exists. The assortative mating I will measure in terms of the probability of mating between two extreme types in relation to the probability of mating of the individual with the same of the same type. Now, if we go to the probability of assortment of 1%, which is quite strong, we actually still see that the distribution is unimodal. But if we go down to 10 to the minus 6 or something, which is quite small probability of mating between the different types of individuals, the extreme types, then we actually see very strong bimodality. But this is in a sense trivial, which is what we expect. This is essentially almost different species, right? So this is not what we expected. Now, if we forget about assortative mating, but instead include genetic incompatibilities, in the model there are actually potentially an individual can have at most 40 incompatibilities, but not all individuals have them, right? And the strength of incompatibility per incompatibility is, for example, 1% or 10% or 20% per incompatibility fitness reduction distribution. And that is to say that we essentially need death of F1 hybrids after the secondary contact in order to produce bimodality, which is again kind of a definition of different species. But if we actually now include both incompatibilities and assortative mating, but reasonably strong, we see that we don't really see with this reasonably strong incompatibilities or assortative mating, still we don't see multimodality. So that leads us to the inversions. We expect inversions to have to act as a barrier to gene flow between the different arrangements, of course, but the question is how effective this mechanism are. So if we now include just inversions and assortative mating, we see that again this is not assortative mating, despite inversions, is not really strong mechanism to assure multimodality. It's not so different from the collinear case. But if we have inversions and incompatibilities, this is actually
Marina, can you pause for a moment? Your sound cut out on me.
I think she's frozen.
Marina, if you can hear us, you've frozen. So I'm gonna stop sharing your screen just so you know. Oh, never mind. That was not me.
Wasn't me either.
Might have been Marina. She just might have dropped.
Oh, I think she did. Hopefully she can get back.
Yeah. She's got 4 more minutes in her session or her talk. If she's not able to join back in and finish in time, then we'll upload her talk separately to the event, so you can watch the recording
Right.
online at a later date. But hopefully she can join back in.
Did seem like her internet was breaking up a little bit.
Yeah, a little bit. I'll shoot her an email.
The joys of a virtual meeting. Her internet must have really gone down.
Yeah. Okay. Well, at this point, there is 2 minutes to the next talk. I've just let Marina know by email.
Yeah. Benoit, do you want to get ready with your slides?
Yes, I will start sharing.
We still got a couple of minutes, but go ahead and share. Good. Is your internet good?
I hope so.
You'll find it. And yeah, we'll just wait the 1 minute to 10:45 before starting. But thanks everyone for your patience.
It's too bad she couldn't let her internet cut out.
You know, it's hard to predict sometimes. My computer froze this morning, which panicked me. No.
Okay, Benoit, why don't you begin?
Okay, so hello everyone. My name is Benoît Perez-Lamarck, and I'm a postdoc in the Institute of Biology of the INS in Paris, and soon to be assistant professor in the University of Toulouse in France. And today I will present some of my work on distinguishing co-phylogenetic signal from phylogenetic congruence to clarify the interplay between the evolutionary history and species interaction. So I will define all these different words like that are in the title. And so during my presentation, I will talk about co-phylogenetics. So co-phylogenetics is the study of the link between the evolutionary history of two interacting clades and their extent interaction. So basically, let's say that we have a system of host and their symbionts. So we assume that we have the phylogenetic tree of the host, the phylogenetic tree of the symbiont, and the extent interaction, so at present. And we try to understand how does this system has evolved. And in particular, so when we try to look at the evolution of these co-phylogenetic systems, we can look at two different kinds of patterns. So the first one, we can try to see whether closely related host species tend to interact with closely related symbiont species. So it's what we call a pattern of co-phylogenetic signal. So it just means that closely related species interact with closely related partner. That's it. Here, like in this example, you can see that the phylogenetics are completely different. And the second pattern we can look at is whether these two phylogenetic trees, or the phylogenetic tree of the host and the phylogenetic tree of the symbiont, tend to mirror each other. So they tend to be more similar than expected. And so this is a pattern of phylogenetic congruence. And so these two patterns, they are quite linked. So for instance, if you have a pattern of phylogenetic congruence, if the two trees are similar, it means that there is also a pattern of co-phylogenetic signal. But actually, like if there is a pattern of co-phylogenetic signal, it doesn't mean that there is a pattern of phylogenetic congruence. And so these two patterns, they have been mixed in the literature in the last decades, and they are pretty important. So it's pretty important to be able to distinguish the two patterns because they can be linked with different processes. So for instance, if we have a pattern of phylogenetic congruence, it can be due to phylogenetic tracking, to co-diversification, when we have like the symbiont, for instance, that will like follow the host diversification and experience co-speciation events, event of transfer, event of duplication or losses. It can be also due to like the successive and simultaneous vicariance events happening at the same time for the host and the symbiont. And it can also be due to preferential host switches. So if we have like the symbiont that diversify by following, like by switching toward closely related host species. So these are the different processes that can explain a pattern of phylogenetic congruence. And the pattern of co-phylogenetic signal, this one is much broader and can be explained by a variety of different processes, such as, for instance, a single event of vicariance can explain like a co-phylogenetic signal, or also trait matching. So if there are some evolutionary conserved traits that are playing a role in the assembly of the interaction, it can also like generate co-phylogenetic signal, so without having like a phylogenetic congruence. And in this context, two different types of method, of co-phylogenetic method, have been developed in order to study these co-phylogenetic systems. And so the first type of method are the called like global fit method. And these global fit methods, they are based on linear algebra techniques. So there are like several methods such as parafit and PACO. And so the global idea is that these methods, they are kind of correlative. So they use like the phylogenetic distances between the host species and the symbiont species, and they try to see whether there is kind of a correlation between these two phylogenetic distances. And so they rely on linear algebra. And the second type of methods are the event-based method. They are like models that try to reconciliate the symbiont and the host phylogenetic trees using events of co-speciation, host transfer, duplication, and losses. And so there are a variety of different methods of event-based methods, such as EMPRESS, that I will focus on during this presentation. And in general, these different methods, so the global fit method and the event-based method, a significant test for either like a parafit, PACO, or EMPRESS are generally interpreted as evidence for phylogenetic congruence, meaning that when it's significant, when the tests are significant, we assume that the phylogenetic tree of the host and the symbiont tend to be similar. But actually, like we don't know whether like they really measure the same thing. So we were wondering, like, do these different method, global fit method and event-based methods, do they really measure the same pattern? And if yes, do they measure phylogenetic congruence only? And so in order to answer this question, we performed simulations. So we simulated different scenarios where we simulated like only like a co-phylogenetic signal, or we simulated scenario with phylogenetic congruence. And for each of these different simulations, we run at the same time global fit method, PACO and parafit, and event-based method like Empress. And so in the first case of a scenario that we consider, it's a scenario where actually like the interactions are driven by trait matching. So we consider that actually the host and the symbiont phylogenies, they evolve like completely independently. So we have two phylogenetic trees that are independent. And we just consider that there are some traits that are conserved, that evolve following a Brownian motion on the phylogenetic tree of the host and the symbiont, such as there is phylogenetic signal in the traits at present. And we consider that at present, species that have like similar traits are more likely to interact together. So there is like a trait matching expression at present. So it generates co-phylogenetic signal because like closely related species tend to have like similar traits. Then we have like closely related species that tend to interact with closely related partners. So co-phylogenetic signal, but no phylogenetic congruence. In the second simulated scenario, we consider just an event of vicariance. And so we consider that interaction at present are only shaped by past biogeography events. So for instance, we consider a scenario where we have like a species of host and symbiont that diversify independently on a single area. And then 10 million years ago, an event of vicariance happened. So the species are isolated in different areas, and they just start radiating, diversifying like separately on the different islands, such that at present, when we look at the host and the symbiont phylogenetic trees, we can see that closely related species tend to be present on the same area. So there is a phylogenetic signal in the biogeographic repartition of the species. And so species of host and symbiont can only interact if they are present in the same area. So it creates a co-phylogenetic signal, meaning that closely related species tend to interact with closely related partners. So the phylogenetic trees are independent. There is no phylogenetic congruence. There is only co-phylogenetic signal. And in the third and last scenario, we consider like phylogenetic tracking, where we have like co-diversification of the host and the symbiont, meaning that actually the symbiont, they diversify on the host phylogeny by experiencing like events of co-speciation, duplication, losses, and transfer. And so it means that actually we can see that the symbiont clade is very similar to the host clade. So there is like a match of the two phylogenies. There is phylogenetic congruence in this scenario, and it's the only scenario that generates phylogenetic congruence. And so for all these different simulations, so we run a lot of simulation, and for each of these simulations, we run global fit methods, such as parafit and PACO, and event-based methods, such as Empress. And we look at the percentage of simulation giving significant tests for these different methods. And so for trait matching and vicariance simulations, so simulations that only generate co-phylogenetic signal and no phylogenetic congruence, we can see that when we run parafit and PACO, we can see that in half of the simulations, we observe significant tests. And when we run event-based method, like Empress, we see 0% of simulation that are significant. Meaning that actually, like this parafit and PACO methods, they can detect co-phylogenetic signal. And when we run, when we had the simulation of phylogenetic tracking that was like the only scenario with phylogenetic congruence, in almost like 100% of the cases, we have significant tests with parafit, PACO, and Empress. So it means actually that these different methods, they are like measuring different patterns. That the event-based method, such as Empress, can measure phylogenetic congruence, so whether like the two phylogenetic trees tend to mirror one another. And global fit methods, such as parafit and PACO, they are not specific to phylogenetic congruence, but they measure like in general co-phylogenetic signals, so whether closely related species tend to interact with closely related partners. And so it's pretty important in terms of processes because like these two methods, they are complementary. So it means that when we run like event-based method, then if the test is significant, it means that there is a pattern of phylogenetic congruence, meaning that it can be due to processes of phylogenetic tracking, of successive and simultaneous vicariance events, or like preferential or switches. And when we run like global fit method, in general, when we have a significant test, it only means that we have a pattern of co-phylogenetic signal, but it doesn't mean like we cannot really say what are the processes that are likely to generate this signal, because there is a variety of processes that can explain a pattern of co-phylogenetic signal. And so we wanted to look deeper into the literature, into recent example of a system where people have used this co-phylogenetic methods. And in particular, in the literature, in the last 2 decades, a lot of papers have been published showing that there is evidence of phylogenetic congruence in the mycorrhizal symbiosis. So meaning that actually they were talking about processes of co-diversification and co-speciation happening in the mycorrhizal symbiosis, so the symbiosis between plants and mycorrhizal fungi. And so we wanted to reanalyze this evidence of co-speciation, co-diversification, and pattern of phylogenetic congruence in the mycorrhizal symbiosis. And so this is a work that has been done by Fantine Baudin this year. So she looked at the literature and she tried to assemble as much as possible of mycorrhizal network with the phylogenetic trees of the plant and the fungi from different publications. So we target different types of mycorrhizal symbiosis and also looking at different spatial and temporal scales. And she ran co-phylogenetic method and event-based method. And by looking at the shape, you can see that there is some evidence of co-phylogenetic signal in the mycorrhizal symbiosis, in the mycorrhizal networks, but there is absolutely no evidence of phylogenetic congruence, meaning that actually closely related fungi tend to interact with closely related plants, but there is no evidence of the phylogenetic tree of the fungi and the plant that tend to mirror each other. So it means that there is no evidence in the mycorrhizal symbiosis of co-speciation or co-diversification happening in these systems. So I would like to acknowledge Fantine Baudin and Hélène Morland, who work with me on this different project. And I would like also to thank you for your attention. I will be happy to answer questions if I have time.
Excellent. So we have a couple of minutes for questions. Oh, there's one in the chat that says, does this research on phylogenetic congruence assume mutual co-speciation between host and symbiont? And does this also apply to plant-eating insects that potentially follow the tree of their host when they speciate?
So does this research on phylogenetic congruence assume mutual co-speciation? So actually, it doesn't really say whether there is co-speciation or not. For instance, a pattern of phylogenetic congruence means that actually the trees are similar, but it doesn't say whether there is co-speciation or not. It's not enough to have a pattern of phylogenetic congruence to say whether there is co-speciation. It can be due, for instance, to having the 2 clades that experience the same events of vicariance, so completely independently, they experience the same vicariance, and so there is a pattern. They have the same phylogenetic history, but there is no really a process of ongoing co-speciation in this system. And about plant-eating insects, that's a good question. We've reanalyzed evidence in the mycorrhizal symbiosis, but if you want to look for other systems such as plant-eating insect, that would be also interesting. I think there is some evidence, of course, of phylogenetic congruence in many systems, but it's just that sometimes the researcher only applied a global fit method such as PACO and parafit, and it's not enough actually to conclude that there is phylogenetic congruence. And so it was the case for the mycorrhizal symbiosis, but I think it's not something general. There is no there's still evidence of phylogenetic congruence in many systems.
Yep. Thanks. We've got just a couple of seconds. Yunfeng, do you want to test your slides?
Yeah, if there are no more questions.
Yeah, go ahead.
All right. Are you guys able to see my slides?
Yes. So let's wait just one few seconds here till it hits 12 o'clock.
Yeah, and I just put in the chat that there's different ways to ask questions. If you want to raise your hand, we'll unmute you. If you want to enter it in the Q&A, that's a good spot too. And then in the chat as well. But sometimes it's hard to track in the chat. So.
And you can direct your questions to all of the speakers in the chat while we're going on.
I like the humming to fill time.
Yunfei, begin, please.
Okay. Hi everyone. My name is Ming Feng, and I've just graduated from Washington University with a bachelor degree in general biology. And today I'm gonna give a talk on my undergrad research, which is about the evolution of Pseudomonas GAC-A-GAC-S gene system under different selective pressures. And specifically, I'm looking at how the GAC mutants and wild type of this bacteria interact with each other. So first, there are two organisms of interest in this study. One is Pseudomonas protogens, which is a gram-negative bacteria that lives in the soil. And the second is a bacterial predator, which is called Dictyostelium discoideum. The name is really long, so I just call it Dicty from now on. And it's a eukaryotic social amoeba that prey on bacteria and also live in the soil. And on the right, here's a graph showing the life cycle of Dicty. And it starts as a free-living single-cell amoeba. And when the food, especially when the food is scarce, it will start aggregate and form a fruiting body, and then finally forming spores to reproduce. And this whole life cycle takes about 7 days. And one thing worth notice is Pseudomonas protogens, like in nature, is like naturally resistant to Dicty's predation. So the wild type is resistant to predation while the mutant is not. And that's sort of one of the major things I'm looking at in this study. And then to introduce the gene system I'm looking at, it's a two-component regulatory system for bacteria secondary metabolism. And the reason why it's so important for this study is as a global system that controls secondary metabolism, it controls the production of secondary metabolites like antibiotics, which is very important for predation resistance. So that also explains why the GAC mutant is susceptible to predation, while the wild type is not. However, the GAC mutant, like they naturally exist in the wild, they can be generated spontaneously. So that makes like me curious, why does this happen? The thing is, there is an evolutionary trade-off there. So predator defense, which sounds like a very cool thing for bacteria to have, is actually very energy consuming. So all those energies and nutrients spent for producing antibiotics can actually be used for just nutrient intake, growth, and reproduction. So that means that although the wild type is resistant to predation, it actually grows way slower than the mutant. Like you could see it on a petri dish, it's growing way slower. That makes me, that brings up two questions that I want to answer. The first one is, how do mutant and wild type interact? And how do mutant exist in nature when predators are likely around? So to test these two questions, I generated 4 hypotheses. The first one is, since previous research have shown that spontaneous mutants can be generated in lab in nutrient-rich liquid culture, I tried to generate it on solid media because that's where the bacterial predator live, and it's also a environment that's more like the soil environment in nature. And second, I proposed that predation by Dicty will significantly reduce the abundance of GAC mutants, while wild type is not affected. And thirdly, in mixture of wild type and GAC mutants, the starting ratio will affect the competitiveness of each strain. And fourth, for the Dicty's predation, it might be a key factor in determining the stable ratio for mutant wild type coexistence. And the first experiment is I try to generate spontaneous GAC mutants. And for the prediction, I think if mutants actually can be generated, it should be having the highest abundance on nutrient-rich media while having the lowest abundance under predation. So to test that, I have 3 groups of setting up with a control, a nutrient-rich environment, and also under predation. And it was all growing under room temperature for 7 days, which is a life cycle of the predator. Then to quantify the mutant abundance, I use a milk agar, which is milk powder mixed with agar. So that is because the GAC system also controls the expression of exoprotease, which eat up the proteins in the milk. So the wild type will form a halo around its colony, as shown here in this picture. So the results for this experiment is I actually was not able to generate any GAC mutants, or maybe it's more precise to say I was not able to detect any mutants, even on the most nutrient-rich condition. And this could be due to just nutrient-rich solid media is not a strong enough selection pressure for spontaneous mutation, or it might be because there are too few mutants to be detected by MuCAGR, since it's pretty coarse technique for detecting mutants. So to keep investigating the interaction between P protogens, GAC mutants, and wild type, we just need to generate a mutant through gene editing and use the artificial mutants to see how they interact. And that leads to my second experiment, which is I generated a GAC mutant using Golden Gate assembly, which is a technique that now called the GAC gene, make it just non-functioning. And in this case experiment, I was trying to test how predation and different starting ratio can affect the interaction of wild type and mutant in the same population. So I tested 3 different starting ratio, which is 1:1, 1:100, and 100:1 under 2 different conditions. One is no predation, one is under predation. And again, it's growing in room temperature for 7 days. Here I uses CFU value as a proxy for abundance, and then competitive index, CI, as a proxy for their relative competitiveness. So to get like individual CFU and CF value of the mutant and wild type, I have to separate them. So I labeled each strain with like a different antibiotic resistant gene, and then use antibiotic selected plates to count their single colonies, as shown here in this flowchart. From the single colony count, I can calculate the CFU value and the CI value. So here's the results. We can first look at the right graph, which shows the mutant abundance. And it's very interesting that the mutant fitness is significantly affected by DICT predation when it's at high or equal abundance with the wild type, or just exclusively mutants, except when the mutant is existing at a very low abundance. And that kind of shows a very interesting behavior of the mutant. It's like cheating on the wild type to survive under predation. Specifically, it's like using the antibiotics produced by the wild type to repel predators. And then if we look at the graph on the left, we can see that for most of the time when the wild type is at a relatively high abundance in the population, it's not really affected by predation. Like even if there's, for the green dots and orange dots, they're basically having the same CFU value. However, when the wild type is existing at low abundance while the mutant is taking the dominance, this population is not really stable. We can see that the wild type's abundance is like really significantly affected. And that might be, there are just so many mutants existing there, and that means a lot of food for the predator. So the predator proliferates really well and starting to affect the fitness of the wild type. And then for the CI value, we can see that firstly, for the orange dots, which shows the results for underpredation, mutant is always less competitive than wild type, no matter what the starting ratio is. So we can see that the existing predator can really control the mutant abundance and keep it at really low, like as a minority in the population. However, when there's no predators around, and especially when the mutant are abundant or at equal ratio with the wild type, it can actually be more competitive than the wild type. And we can see that from here, it has a CI value way over 1. And that means a relatively higher competitiveness for the mutant. So finally, the conclusion is predation controls GAC mutant abundance through a negative frequency dependent process. And it's definitely not the only process controlling this, but it's one of many potential factors. And here on the right, upper right, here is a graph showing how the negative frequency dependent process works. is when the population starts with a high abundance of mutants, the predation can just control the mutant abundance and reduce it to a relatively low ratio and just keep it stable in that wild type versus mutant ratio. And this phenomenon also can explain why high mutant abundance does not exist, since the predation can just eat up all the mutants. However, more experiments are needed to explain why, like in predator-free environment, mutant does not always take the dominance. Like it's the CI value on the previous slide shows that it's taking dominance when mutant are abundant, but not really when the mutants start at like relatively low ratio. So thirdly, in nature, it's unlikely to find environment that are free of both predators and competitors. So that could be why gap mutants do not take over the wild type, which is the natural communities in the wild, even though it's like exists spontaneously in nature. So the third conclusion is has potential application in biocontrol because Pseudomonas species are known for having symbiotic or cooperative relationship with plants. And that means if there are predators around in the soil, it can make sure that there are along the roots of the plants, only the beneficial wild type is existing there. And the wild type is beneficial because it produces antibiotics that repel other plant pathogens. While if they're all mutants, as shown here, it's not going to be helpful because no antibiotics are produced and the pathogens can easily infect the plant species. Yeah, and that's all I want to share. Thanks for listening.
Excellent. We have time for a couple of questions if anyone has some. Just raise your hand or type it in the chat or in the Q&A. Well, I'll ask a question. What first attracted you to this particular gene and looking at the interaction within this community?
I would say it's there's another person in our lab worked on this gene before and on different strain, and he tested that if you delete that gene, this mutant is susceptible to predation. So I already know this phenomenon, but like I don't know why like the mutant exists in nature and we don't really know how they interact, especially under predation. So I was trying to use the mutants and do some kind of behavioral experiment looking at the interactions between them.
Good. Anyone else? Good. So why don't we, Nami, why don't you get your slides up? We still got a few seconds to wait.
Does that look fine?
Excellent.
Awesome. Let
Let's
me
wait.
know when to start.
Will do. Wait
Thank
just
you.
a couple of seconds here. Got about a minute and a half. Oh, a minute. Remember, you can type questions to the presenters in the chat or Q&A if anything comes up for anybody.
And in the meantime, while we wait, I'm going to try, for those of you who want to catch the end of Marina's talk, she's going to give it again today in the, which talk session is it? I don't know the talk of the session, but later today, and I'm going to adjust the schedule right after this Marina, do you know what the session's called? Genomics. Okay, in the genomics session later tonight.
Nanami, please.
Awesome. So hi everyone, I'm Nanami, a PhD student in Von Cooper's lab at the University of Pittsburgh. And today I'll be talking about my thesis project on how the emergence of tutor phages lower both bacterial and phage population fitness. So before I begin, I just want to do like a background intro on phage. So phages are bacterial viruses, and some of them can infect bacteria and integrate their genome into the bacterial chromosome. And when that happens, as bacteria divides, the phage genome is also copied. And because of this, bacterial fitness is also tied intimately to phage fitness and vice versa. Sometimes phages can also bring beneficial genes with them and affect host fitness that way. And the relationship phages have with bacteria can shift once phage becomes induced. So I work with Pseudomonas aeruginosa, and many Pseudomonas aeruginosa strains actually carry PF or Pseudomonas filamentous phage in their genome. And filamentous phages are unique from your traditional phages in the sense that they don't require host cell lysis or host cell death in order to leave the host. They're also quite important in the context of bacterial infection. So what I mean by this is Pseudomonas, these PF phages can, because of their physical property, they can form these liquid crystalline structures, and these structures can wrap around the host cell, and it acts as a physical barrier that helps shield the host from the environment, such as drug. And so hosts with such structures can tolerate higher levels of drug. Having such structures in sites of infection can also delay wound healing, make it harder to wash away bacteria. And in the context of cystic fibrosis, older patients who are chronically infected with Pseudomonas aeruginosa also tend to have bacteria that are positive for these PF phages. And these PF positive clinical isolates tend to also be more drug resistant. So we could see that PF phage is quite important in the context of Pseudomonas aeruginosa infection. So what our lab does is a lot of experimental evolution, which is a very powerful tool to study adaptation. And what we do, at least in the context of my study here, is we can take a clonal isolate of our ancestor, Pseudomonas aeruginosa, and seed starting population in cystic fibrosis media, which contains nutrients that you would find in a cystic fibrosis lung. And we can passage this over the course of 12 days and compare our evolved population against the ancestor. And this evolution experiment acts as a forward genetic screen. So we can identify mutations and genes that are important for the bacteria adapting to such an environment. And our lab has published in the past to show that nutrient availability can select for clinically relevant adaptation. So for today, I won't be talking about this study. I'll be talking about a new discovery we made, which was a mutation within the PF repressor gene. So the PF repressor gene is within the prophage genome. And what that means is not only does the phage have this mutation, but so does the bacteria because the phage is integrated into the bacterial chromosome. So when the phage repressor gene is functional, the phage remains within the chromosome. But once we have a mutation that turns off this gene, we start seeing phage excision, replication, and it turns the bacteria into a phage factory, and we start seeing these hyperreplicative phages. And we can see this when we whole genome sequence our bacteria. So this is a read depth graph of our ancestor strain. And notice how the number of reads mapping to the phage region is relatively the same as the genes flanking it, the bacterial genes flanking it. So anything within the red bars is the phage genome. But once you have a phage repressor mutation, we start to notice a ton of reads mapping to just the phage genome, which indicates that every bacteria has more phage copies in the cell, and that these are hyperreplicative phages. What's interesting then is that once we have a ton of replication happening intracellularly, we also notice the presence of smaller phage genomes. And what we think is happening is these full-length hyperreplicative phages are cooperators that help produce viral proteins such as capsid. And these capsid proteins are public good in the sense that the phage making the capsid proteins aren't necessarily the phages using the capsid. Other phage can also use the public resource. On the other hand, we start to see these cheater phages with smaller genome sizes, so these mini phages that don't have capsid genes. And so because of their smaller genome size, not only can they replicate faster, they also don't contribute to the public good, but they can shuttle their genome into the phage capsid, thereby cheating off of these full-length cooperative phages. And again, whole genome sequencing can show us the lack of capsid genes in our phage population. And I normalize these phage copy numbers by the bacterial chromosome. And we can appreciate that once you start seeing these mini phages, these cheater phages, the number of phage copy also goes up. So every bacteria has more phage once you start seeing the emergence of cheating. So then our question was, why was this selected for? So I did growth curves, and interestingly, somehow the ancestor actually grows better than our mutants, our full-length infected bacteria, as well as our full and mini phage infected bacteria. So again, why was this selected for if there's a growth disadvantage for making phage? So we did competition experiments where we co-cultured our ancestor against our mutants here. And before I show you the data, I'm just going to walk you through how to read this graph. So anything positive means that the mutant outcompetes the ancestor. Anything negative means that the ancestor outcompetes the mutant. And I also varied the starting frequency of the competition such that anything closer to 0 means that the mutant's rare in the starting culture, and anything closer to 1 means that the mutant's common. And for the full-length infected bacteria, it can relatively do better against competition against ancestor. But once the bacteria has both full and mini phages, it decimates ancestor to the point where you could start with a starting culture that has 0.1% of this full mini infected bacteria. And by the end of 24 hours, ancestor bacteria is barely detectable. So in order to understand what is going on, I took time points and tracked bacteria numbers over time. And so we can see that ancestor by itself has a higher yield, but once you co-culture ancestor with our full-length infected bacteria, that lowers the yield. And once you co-culture with full and mini infected bacteria, the full mini bacteria can actually suppress ancestor early on during competition and then outnumber it, thereby giving it a higher relative fitness, a competitive advantage under competition with ancestor, despite the fact that producing a bunch of phage has a growth defect. So then my next question was, is it the bacteria that's responsible for this pattern that we see here, or is it the phage responsible for this suppression? So to do so, I filtered out the bacteria, so we're just left with supernatant, which should contain our phages, cultured that with our ancestor and do growth curves again. And we can see that, yes, our full-length infected full-length phage does suppress ancestor, but once you have a combination of full and mini phage, it can suppress it early on. And this shows that our phages are responsible for the early growth suppression that we see in the ancestor. And given this data, as well as the higher copy number in our full mini bacterial whole genome sequencing, we think that the bacteria with both full mini phages are just making a bunch more phage than our only full infected bacteria. And that's why having both full and mini allows the bacteria to outcompete ancestor better than if it just had the full length phage. So going back to my read death, we see our cheater emergence. What's interesting, though, is subsequent overnight cultures of these bacteria leads to phage extinction. And what we think is happening is as the bacteria divides, the daughter cells don't inherit both full and mini copies. They can inherit only mini phages by chance, especially if you have a proportion skewed towards mini phages. The chances of the daughter cells just inheriting mini phage goes up. And once you have a bacteria that's only infected with these cheater phages, it becomes a tragedy of the commons in a sense that these bacteria, one, they can't make phage, the phages can't cheat off of other phages, and over time you lose the phage, and the phage goes extinct. So to just recap, we had our ancestor, and after 12 days, we saw this repressor mutation get selected for, which allows hyperreplicative phage to increase in number, as well as allow the bacterial host to outcompete its ancestor. But then this allows emergence of cheating in the phages, but also this mixture of full and mini allows the host, bacterial host, to also outcompete other bacteria. But this state is very unstable in the sense that over time, through uneven vertical inheritance, you'll get daughter cells that don't have the full length phage, and that leads to extinction. And once the bacteria loses its phage, it can no longer outcompete ancestor. And we can imagine
3 minutes.
Thank you. We can imagine the cycle repeating once we see reinfection of our ancestor phage. So before I finish, I want to give my acknowledgements to my lab, my PI, Von Cooper, as well as the Cooper Lab and lab alumni, especially Shelley Scribner, who I've inherited this project from, my funding sources, SSE, as well as the organizers of this conference. And happy to take questions. Thank you.
Excellent. So we have time for a couple of questions. If anyone has one? No? Well, I wonder about just the basic biology. I've never heard of this kind of phage before. How do they assemble the capsid in the virus particle without actually lysing the cell?
Yeah, so what's interesting is that instead of popping open the cell like your traditional lysis lysogeny phages that you learn in the textbook, these phages can actually secrete the phage capsid out. So that doesn't require the host cell lysing in order to leave the cell.
Oh, never heard of that.
Yeah, so like M13 is a famous example of filamentous phages. Also, your cholera CTX5 phage is also filamentous phages. So they're out there, but I don't know. I guess less people study them than your other phages.
There's a question in the chat from David Baum that says, I may have misunderstood, but it seems ironic that path to getting cured of phage is to lose a phage repressor gene. Is that right?
Yeah, so losing the phage repressor gene leads to hyperreplication. you're no longer repressed, so you can make a bunch of phages, but then you have this very unstable state in which not only is cheating then become the strategy that is advantageous, but then you also have the dynamic with the host and cell division that then if you have a very high population that's very skewed towards like more cheater phages than your full length phages, you can lose your full length phages through cell division. So that's what we see, at least in a very well mixed culture. I have done cultures that have spatial structure, in which case you can see coexistence of and possibly these phages can stay in the population and not go extinct. But at least in my system where I'm using liquid, we do see loss of phage.
And there's one more question. I'm sorry if I missed it, but how do the bacteria with active phages suppress the ancestor?
So we believe, so I didn't have time to show this and given the time constraints, but we do know that just having a bunch of phage in the environment is very stressful. You can have a bunch of phages infecting your ancestor at once, and that can induce cell stress and cell death. So that's one way a mutant can use their phage almost like a bioweapon against their ancestor. And then these mutants are also a little bit more phage resistant, so they can tolerate higher levels of phage in the environment. So let me see if I answered that question. So yeah, cell stress from just having a bunch of phage in the environment can lead to cell death.
Great. So we're at time now. So everyone move on to your 1:30, whichever way you're going. We can hang on. We'll hang on a few minutes if somebody else wants to ask the presenters more questions. If not, email them directly.
And I think there is a question that I think has not been answered yet in the Q&A.
Oh.
Can you for
Oh, there you go. What question about this cheating system are you interested in investigating in the future?
So one, I'm interested in whether we could use these cheater phages as sort of a gene drive in order to basically cure these bacteria of their PF prophage. We do know that these PF phages are important in the context of bacteria infection. So if we can use and engineer it in order to cure it, perhaps these bacteria will become more sensitive to antibiotic treatment. So that's like a long-term, like hopefully someone else or someone can do it in the future, but that's like a long goal. And I believe there's another question. How many phages are known to infect PA? I don't know. And I think the number is actually, we haven't, because phages, we have a lot of unknown phages out in the environment. Estimates are probably on the lower end than what we actually know. But yeah, awesome. Thank you.
Thanks. Well, thanks, everyone.
Thanks, Mark, for hosting. I think we've got a gap now of an hour until the next sessions start, and then there's a couple workshops over the lunchtime hour, but otherwise take advantage of your break. And thanks very much, speakers. They were all really great talks.